Hello, my name is Bob Goldman, and I'd like to speak to you today about intermediate filaments. Intermediate filaments are found both as complex networks in the cytoplasm and in the nucleus of vertebrate cells. The naming of intermediate filaments may be of interest to some of you, as the name is derived from the fact that they are midway in size between microfilaments and microtubules. So they have a diameter of 10 nanometers. So they were originally called 10 nanometer filaments, but now they're called intermediate filaments. The intermediate filament protein family is complex and in fact is encoded by one of the largest families of genes in the entire human genome. Typically, intermediate filaments have a highly structurally conserved central rod domain, here seen mainly in red. And there are 5 major types of intermediate filaments. The first 4 here, types 1 through 4, are found in the cytoplasm. So those are the cytoskeletal intermediate filaments. The characterization of filaments structurally is based on their as I mentioned, highly conserved central rod domain, which is separated by these linker groups called L1, L1, and L2. And these separate the central rod into segments 1A, 1B, 2A, and 2B, which are all highly alpha helical in nature. The keratins, which are the first 2 types, are both the acidic keratins and basic keratins are complex because they form heterodimers as the basic building block. And these heterodimers then interact to form, ultimately, 10 nanometer filaments. There are presently known to be over 50 keratin genes in the human genome. In the case of the type 3 intermediate filaments, such as Vimentin, desmin, peripherin, and glial fibrillary acidic protein, these form homopolymers. And in fact, Vimentin will be the major topic for today because it's the simplest type of intermediate filament protein, having only one protein chain which self-assembles into 10 nanometer filaments. And then the type 4 intermediate filaments, the last type in the cytoplasm, consist of neurofilaments, which are actually 3 protein chains forming a complex mainly found in the nervous system, nerve cells of the brain and the spinal cord and the peripheral nervous system, and a protein called nestin, which is expressed mainly during early development, cinnamon and alpha-nexin, which are other forms of intermediate filament proteins, which also form complex polymers. And finally, there's the type 5 intermediate filaments, which are the nuclear lamins, which we will discuss in the second part or in the second lecture. So that within cells, there's a remarkable complexity of intermediate filament networks. Almost every cell type that one looks at has a different, not only intermediate filament composition, but also intermediate filament network formation. You can see on the left a Vimentin filament network in endothelial cells with a ring of filaments around the nucleus. And then on the right, you can see keratin in an epithelial cell, and you can see this remarkably complex network radiating all the way from the center of the cell, where it surrounds the nucleus as a cage, all the way to the cell surface. Now, the functions of cytoskeletal intermediate filaments are numerous, and we won't have time to discuss all of them today, but we'll discuss a few of them. But let me summarize by saying they're very important in the mechanical integrity of cells. They're very important in the shape of cells. They're involved in both shape determination and maintenance. They're very involved in cytoskeletal crosstalk and stability. That is, they frequently bridge between microtubules and actin filaments. They're involved in signal transduction, adhesion, both cell-cell adhesion and cell extracellular matrix adhesion. They're involved in cell motility, and they're involved in the positioning and tethering of organelles within the cytoplasm. So let me just briefly go over the structural features of intermediate filaments and how they're built in the cell, how they're assembled. So the typical intermediate filament is made of dimers, so two protein chains. For instance, in the case of Vimentin, since it's a homopolymer, it makes homodimers, and two dimers interact to form head-to-tail assemblies, which are in exact parallel and in registers. So the central rod domains actually line up precisely so that they form a coil-coil, and then we have the tail at one end and the head domain, or the N terminus and the C terminus at the opposite ends of the rod, of the dimer. And then we can see this at just a blow-up of this type of schematic, and you can see that so-called helix 1 and helix 2 are separated by this region in the center called the linker 1-2 region. And then we have the C terminus, which is relatively unstructured, and then the N terminus at the other end, which is also relatively unstructured in the case of cytoskeletal intermediate filaments. During polymerization of, in this case, human Vimentin, or any form of Vimentin, because it's a highly conserved protein, we can see that in the far left panel, the formation of dimers, and the two globular regions on one end of the dimer actually are the C-terminal domains. And if you then go the next step in assembly, and this is all done in vitro using bacterially expressed protein, you assemble what are called unit length filaments. These are actually dimers which interact with each other, and there are 8 of them. So there are 16 dimers which form a 32 protein chain unit length filament. And these unit length filaments, which are approximately 50 nanometers in length, actually interact with each other in tandem to make longer and longer filaments. There's 2 interacting at the lower left side of this slide during the assembly process. And then finally, they go, once you get long linkages of ULF, you then go through a radial compaction, which you can see in the last panel here. And then it can be seen even in a more schematic way in this next slide, where you have unit length filaments interacting with each other, linking in tandem to form longer and longer intermediate filaments, which then go through a super compaction to form mature intermediate filaments. The other important point about filament structure is that it's apolar, because the dimers, which are in parallel in a register with respect to the 2 protein chains, interact in a head-to-tail fashion, which is called longitudinal assembly of dimers, and they also interact laterally in an oppositely oriented and staggered way, so that you wind up getting tetramers with opposite polarity. And building up this structure into a 10 nanometer filament gives you what's called an apolar filament, because it doesn't have a plus end and a minus end like microtubules do, and like actin. So now I'd like to go on and just tell you how we prepare intermediate filament proteins from cells. And essentially, if you look on the left panel, it just shows you a blow-up of a region of a cell which was fixed and stained with antibody. It's a fibroblast, and you can see the complex Vimentin network. In the middle panel, this cell has been extracted with a solution containing high salt, detergent, and DNase-1. The DNase-1 is used to degrade chromatin. It also helps to remove actin from the preparation, and there are no microtubules in these preparations. And if you look on the next panel, you'll see that at higher resolution, higher magnification in the EM, that in the electron microscope, that in fact these are intermediate filaments, and there's virtually no other structure in the cytoplasm of these former cytoplasm of these cells. And on the right panel, you can see the major band, which is Vimentin, and just above that, there are the nuclear lamins, which we'll speak about later or in the next talk. And finally, at the top of the gel, there are several proteins which are associated with intermediate filaments, such as nestin in this case, and also a protein called plectin. Because intermediate filaments are so easy to isolate, and in fact, because they are pelleted at low speed, and almost virtually all of the protein, intermediate filament protein in cells like fibroblasts can be pelleted at low speed, and virtually no protein is detectable in the supernatant. For many years, intermediate filaments were thought to be very stable polymers inside of cells. Another property that led to their, or leads to the, I think, misconception that they are very stable is the fact that in an old paper by Paul Janmay many years ago, he demonstrated that under conditions of stress and strain in vitro, in fact, intermediate filaments remained undamaged, as you can see in the panel on the left, whereas microtubules and actin filaments ruptured at relatively low shear stress in this preparation. On the right side, you can see a drawing from a recent article in the literature which shows that intermediate filaments, in addition to being undamaged by high shear, can actually be stretched up to 3.5 times their normal length. And once they become stretched to this extent, they become very difficult to break. So in fact, this is an atomic force microscope depicting what happens to an intermediate filament when you stretch it. And basically, this is called, their properties are known as strain hardening properties. And for these two reasons, both the fact that they're very extensible, they can become very rigid when stretched, and that they seem to be very insoluble in biochemical preparations, they were thought for many years to be less dynamic than their cytoskeletal counterparts. So let me go on and tell you a little bit about the dynamic properties of intermediate filaments. So when we turn to live cells, we realized that intermediate filaments are very dynamic in vivo. And in fact, if you do a photobleaching experiment where you look at recovery of photobleaching, the FRAP analysis, you can see on the left side a cell that was microinjected with rhodamine conjugated Vimentin. And then in the next panel, B, immediately afterwards, that fiber was bleached, photobleached. And then in C, D, and E, you can see the photobleach recovery coming back. And if you analyze in the bottom panel the recovery of fluorescence, you'll find out that it's equal all along the length of the fiber as it recovers, which indicates that, in fact, the filaments are not polar, that they can actually exchange subunits all along their length. Another aspect of intermediate filament dynamics can be seen very dramatically when cells are attaining a shape in culture. So if you take trypsinized cells, which are rounded up, placed in suspension, and then place them down onto a glass cover slip or slide, and then watch them during the spreading process by fixing and staining at different times, up to 3 or 4 hours, you will see in the top slide a lot of very small spots or particles. And these are enriched in Vimentin. These cells were fixed and stained with Vimentin antibody for immunofluorescence. In the middle slide, you can see, in fact, that as the cell spreads, small filaments or short filaments, which we call squiggles, form. And then these squiggles appear to link end to end, as we saw in vitro, to form longer and longer filaments until a fully formed mature network is formed in spread cells. Movies of this reveal how dynamic this process, in fact, is. This is a later stage of spreading, but if you watch the particles in the movie and the short filaments or squiggles, you'll see that they're very dynamic. They're moving bidirectionally towards the edge of the cell, and they can both move towards the nucleus and away from the nucleus, as you can see. Now, the types of movements, the speeds can be up can be very fast, up to 1.5 to 2 micrometers per second. This indicated, of course, that they were probably moving along structures like microtubules. And in fact, those Vimentin particles, especially moving at high rates of speed, can be co-localized when you fix and stain using double immunofluorescence. And you see here microtubules in the middle in red and Vimentin on the left in green during early spreading. And then the overlay on the right, on the upper set of pictures, you will see that that there's a very nice overlap between the Vimentin particles and microtubules. And in fact, if you look on the bottom two panels, you'll see that most of the particles co-localize with the molecular motor kinesin, which of course is a microtubule-based motor. And without giving you all the data, I just want to summarize by saying intermediate filament particles move bidirectionally because, in fact, they're associated with both dynein and kinesin, so they can move both towards the plus end and the minus end of microtubules. And they move at relatively high rates of speed. They seem to slow down when they form filaments, and they continue moving until filaments meet each other, short filaments meet each other, and then become longer filaments. So mature IF seem to be made from particles, squiggles, linking up to form long IF, and depending on microtubule-based motors, and in fact there's evidence for microfilament-based or actin-based motors participating in this process. Now that I've demonstrated that intermediate filaments are dynamic, let me go on to say that intermediate filaments are also very important in cell shape. And this is no better depicted than in the epithelial to mesenchymal transitions that take place both in early development and in the conversion of tumor cells to metastatic tumor cells, or benign tumor cells to metastatic tumor cells. And these changes in cell shape are always accompanied by an upregulation in Vimentin expression. Typically, epithelial cells, which you see on the left, express keratin, and when they go through the epithelial mesenchymal transition, they begin to express, and eventually Vimentin is expressed at very, very high levels. So Vimentin expression is a hallmark of the cytoskeletal changes that take place during this EMT. On the next slide, we can show that Vimentin actually can induce changes in epithelial cell shapes very rapidly. So if we look at the top panel, we can see an MCF7 cells, which is a human breast epithelial cell, which is round, it's a single isolated cell. And then in the middle panel, it's a cell just observed 2 hours after it was microinjected with bacterially expressed Vimentin, and then fixed and stained. So in a very short period of time, the cell changes its shape just due to the expression in the form of protein injection into the cytoplasm of an MCF7 cell. Eventually, that cell will recover its rounded shape because, in fact, Vimentin expression can't be sustained. On the bottom panel, we can see Vimentin on the left and the result of silencing Vimentin expression using shRNA. And you can see the cells that are silenced by the green color, and which is using GFP as a marker in this case. And you can see the cells that are not transfected, in fact, are epithelial, are normally shaped like a mesenchymal cell. And the ones that are transfected and silenced, you can see have changed their overall shape to an epithelial cell. So it looks like both the reverse of the EMT, the mesenchymal to epithelial transition, and the EMT itself can take place just by controlling Vimentin expression. So cell shape seems to be very dependent, in this case, on the intermediate filament composition of cells. I should mention that microtubules and actin are not affected in these experiments. now I just want to mention that not only do you get shape changes in epithelial cells when you put Vimentin or you express Vimentin in them, but Vimentin also increases epithelial cell motility. So here we see in the upper left panel Vimentin being expressed in an epithelial cell colony. This is an MCF7 colony, which you can see in phase contrast below. And this is just a few hours after transfection. You begin to see expression of Vimentin, as you can see in green. And 24 hours later, you can still see Vimentin networks in these epithelial cells, but now all of the cells in the lower right panel have moved away from the colony's edge. So this tells us that Vimentin expression will alter the adhesive, the cell-cell adhesive properties, the cell substrate adhesive properties, change the shape, and cause the cells to migrate away from the colony. Again, showing the importance of Vimentin in motility. And finally, I just want to emphasize the requirement for Vimentin in motility by looking at, on the left, normal Vimentin-containing fibroblasts. In the middle, these are fibroblasts which have no Vimentin. They're not, in fact, they don't express any intermediate filaments. They're from the original Vimentin knockout mouse. And if you look, you can still see in those cells in the middle lots of activity around the surface. These are surface ruffles, but no directional movement. They can't move anywhere. And you can see on the bottom of each of these movies, you can see the tracks made by the cells over a 10 to 12 hour period. In the right panel, all of the cells are normal, except for this one, the tip of my finger, which in fact is a cell that's expressing a dominant negative mutant of Vimentin, which is seen at the top. It's basically a small piece of Vimentin which contains the initiation site of the coil domain plus the N-terminus. And this acts to disassemble filaments in cells. And you notice that one cell that's expressing this mutant protein is the only one in the field, virtually, that doesn't move, as you can see by the tracks below. We then decided to look at Vimentin intermediate filaments in moving cells. What do they look like? So we just take moving fibroblasts. In this case, it can be mouse fibroblasts or human fibroblasts. They look virtually indistinguishable. And you can see in the top left the typical fan-shaped moving fibroblasts. You can see only now looking at Vimentin, you can see Vimentin particles are present in the lamellipod region, and at the leading edge, the lamellipodium contains, in the upper right panel, you see lots of closely packed Vimentin particles, but filaments only seem to be forming or assembling in the rear region of the lamellipodium. But the protein is certainly there in a disassembled state. Below, in the top panel, top three images, you can see a serum-starved fibroblast with Vimentin filaments radiating all the way out to the edge. And then it's very easy to induce ruffling in such cells, as has been done for many years in many laboratories, just by adding serum back. So you take serum-deprived cells. The cells were starved for 48 to 72 hours. Vimentin typically goes all the way to the surface. The cells don't move. They're very quiet. But as soon as you add, actually within 10, 15 minutes after you add serum back, on the bottom panel you can see the rapid formation of lamellipodia, and you can see rapidly that the same configuration we see in normal moving cells, we can now see in these serum add-back experiments, where you can see Vimentin particles enriched in the leading edge, and then a sort of a gradient of filament formation back towards the nucleus. Then we asked the question, does the disassembly and retraction of Vimentin intermediate filaments following serum addition involve phosphorylation? And to do this, we used a phosphospecific antibody, which we received from the Inagaki lab in Japan. And you can see in the top three images on the left, Vimentin on the left is stained with Vimentin antibody. In the middle, it's an antibody against Phosphoserine 38, which is in the N-terminal domain of Vimentin. And on the right is an overlay, which of course just gives you the same pattern. On the bottom, we can see a cell which was fixed and stained within a very short period of time, within a couple of minutes after we added serum back. And you can see now in the middle panel, Vimentin is on the left. The middle panel shows what the serine 38 phosphospecific antibody looks like. And you can see now that the entire cell lights up, is fluorescent. All these are indirect immunofluorescence images. On the right is the overlay. But blow up of the region in the seen in the first panel, panel D on the left, is seen over on the right side here, where you can see that actually it looks like the Vimentin is retracted back from the surface and that it's left behind particles and short filaments. And here we can see in an SDS acrylamide gel the result of, and a Western blot, that in fact phosphorylation within 1 minute after adding serum goes up very significantly, many fold. If you look at the 0 time versus 1 minute, you'll see the Vimentin is rapidly phosphorylated. In fact, it only takes seconds to become hyperphosphorylated under these conditions. So this suggests that Vimentin phosphorylation is involved in the disassembly, local disassembly and retraction of filaments away from the cell surface in these regions. And I should mention that these are the regions which seem to initiate lamellipodia formation. We also used another trick to look at Vimentin phosphorylation, and that is using a form of the small GTPase signaling factor called RAC1, which is a member of the Rho family of GTPases. It's very important, we know from the actin literature, that this particular signaling molecule is very important in reorganizing actin in the lamellipodial region. And to do these experiments, we used the photoactivatable form of RAC1 provided to us by Klaus Hahn from the University of North Carolina. And this photoactivatable form of RAC1 will become active only when you shine 458 nanometer light on it. And when we expose cells which are expressing this to global irradiation, that is, we just irradiate the whole cell or a whole culture dish, you can see that, in fact, the intermediate filaments seem to retract back away from the cell surface under these conditions, as in fact lamellipodia form. And this is accompanied, again, by a huge increase in the phosphorylation of serine 38. So normally you grow the cells in the dark, and when you shine light on them, they activate this RAC1. And RAC1, it turns out, binds to PAC, and PAC is a kinase which is known to phosphorylate serine 38 in the Vimentin head domain. On the upper left, we see the result of a local irradiation experiment with Klaus Hahn's PA-RAC1. This cell, if you look beginning now, was serum starved, and then we photoactivated that small region which you see in the movie. But the important thing here is to look at the edge of the cell, and you'll see that when the Vimentin pulls back, now the ruffle, in fact, forms. So Vimentin seems to disassemble and retract from the cell surface. And you can see this even in regions which have very thick cables of Vimentin near the cell surface. In the second upper panel, you can see actually it looks like a steel cable unwinding after photoactivation of RAC in this small region. And as this unwinds, you can see the formation of short filaments and squiggles which seem to spin off from this cable. And it's only then that we really start to see active lamellipodia forming. So in both cases, it looks like Vimentin actually is stabilizing the cell surface, and only when you disassemble and dismantle it, presumably by phosphorylation and retraction away and disassembly and retraction away from the cell surface, do you get lamellipods forming. Now we did one other series of experiments to show the importance of Vimentin, and that is using a peptide called the 2B2 Vimentin peptide. 2B2 is a mimetic peptide which is derived from the 2B region of the central rod domain. And with our colleagues Harold Herman and Uli Aby, we discovered that in fact if you expose Vimentin filaments on the left side, panel A, to this peptide and leave it for a relatively short period of time in vitro, this is an in vitro experiment, you disassemble intermediate filaments into these short pieces, which in fact are unit length filaments. So using this 2B2 peptide, we carried out some microinjection experiments. In the top panels here, just a repeat of what the peptide does to intermediate filaments, disassembles them into small pieces, which in fact are unit length filaments. And if you look at cells following injection of this peptide, you see on the lower left, figure D, a cell which was injected, which had been grown which had been grown in the absence of serum. You can see that in fact after injection, the filament network is retracted back towards the nucleus, and there are many particles and short filaments in the region between the nucleus and the cell surface. And in fact, you also induce lamellipodia to form, as seen here by one of the leading edge markers, ARP23, which is an actin-associated protein. And here we see a control cell, which was injected with scrambled peptide, and you can see that there's no retraction back of the network, there's no disassembly. In the next slide, I just want to show you a movie of a cell which was injected with a relatively high concentration, 2 micrograms per mil of the 2B2 Vimentin mimetic peptide, and the filaments retracted. This was a GFP-tagged Vimentin expressing cell, and you can see that the filaments were retracted back towards the nucleus, and then we in the absence of serum, we get basically uncontrolled ruffling all over the surface. And this cell cannot move anywhere, it just sits there. So this fits nicely with our Vimentin null cells, where we saw ruffling all over the surface. In this case, all we did was to inject the 2B2 peptide and in the absence of serum. And then we did another experiment using very low concentration, 0.5 micrograms per mil in the injection buffer. And this cell was injected just to the left side of the nucleus. And this cell was in low serum, and it was not, it was 1.5% serum, and it was not moving at all, and there was no obvious ruffling. And we injected just to the left of the nucleus. And when you do this, you see at the point of injection, very close to that region, we induced a ruffle. And if you look carefully, you can see that this cell even begins to migrate in the direction of the ruffle up towards the left of the screen. So in summary, these observations tell us that Vimentin intermediate filament assembly and disassembly are required for the proper mesenchymal fibroblast type cell locomotion. The regional and local disassembly of intermediate filaments into their structural building blocks, both the particles and squiggles, modulates the formation of lamellipodia. And conversely, the regional and local assembly of intermediate filament networks appears to be involved in inhibiting the formation of lamellipodia. And what this means to us is that it actually acts as a brake or a stabilizer to the cell surface. Intermediate filament disassembly and assembly appears in turn to be regulated by kinases and phosphatases, many of which are involved in signal transduction. I'd like to acknowledge and thank all of the members of my own laboratory who have contributed in recent years to these studies, and our collaborators both here and overseas, and especially to our granting agencies, the National Institute of General Medical Sciences, the National Cancer Institute, and Hannah's Hope Foundation for supporting our work. Thank you.